So we are moving towards the end of this beautiful day, and so we are now having our last presentation, which is the winner of the Best Laboratory-Based Abstract Award. The presenter is Irma Airoldi, research scientist at the Gaslini Institute in Genova. Irma is a old friend, colleague, collaborator, expert in gamma delta T cells and not only. And I am delighted to introduce her. Her talk, a novel advanced therapy medicinal product composed of activated gamma delta T lymphocyte and NK cells to counteract leukemia and relapse in pediatric patients transplanted with alpha beta T cell depleted haploidentical transplant. Thank you, Irma.
Thank you, Alicia, for your presentation, and thanks for this opportunity, of course. Now I will talk about the generation of this novel ATMP, so a novel advanced therapy medicinal product. As Alicia told you, this is composed of activated gamma delta T lymphocytes and NK cells. So the first question is, to move the slide. Okay. Why gamma delta T lymphocytes and NK cells? Because these cells have some unique and multifunctional properties. In fact, they are both killer cells that bridge between innate and adaptive immunity. And most important is that they recognize antigen in MHC independent manner, thus avoiding the possibility to induce GVHD once infused into the recipient. They may kill target stress cells, so including tumor cells and viral infected cells, through a wide panel of receptor and ligands. And they shared also killer activating receptors, such as NKG2D and A, and both produce interferon gamma in response to antigens, and may mediate ADCC thanks to the surface expression of CD16. Another important issue is that gamma delta T lymphocytes and NK cells may develop into memory-like cells. So for all these biological features, gamma delta T lymphocytes and NK cells have attracted high interest as potential tool for adoptive cell therapy. So after a series of experiments in our R&D, we have set up the production process in our manufacturing GMP facility. The process takes 14 days, and it has been performed in a closed system thanks to the Clinimax Prodigy platform and the use of biobag for cell culture. We also used all reagents that were clinical graded. Our starting material was represented by leukapheresis that had been obtained from adult healthy donors. Apheresis has been subjected to a Fickel gradient in order to obtain mononuclear cells that have been stimulated using zoledronic acid and IL-2 for 4 days. At day 5, cells were then expanded and cultured again in the presence of IL-2 alone, and then at day 14, we have performed an alpha beta T cell depletion. So we have obtained the final cell product, but we wanted to produce an ATMP ready to use. So we have cryopreserved our cell product in order to have cell product really promptly available to infuse into the patients under the request of the clinicians. All this process has been followed by a series of quality controls in accordance to the analytical methods of the European Pharmacopoeia or other analytical methods that have been validated in our QC lab. At the end of the process, the ATMP has been checked for cell count and viability, for phenotype in order to understand the purity and impurity present in our cell product, for safety, so sterility, the presence, or better, the absence of mycoplasma and bacterial endotoxin, and for potency, so the biological activity of our cell product that in our case was represented by the cytotoxic activity against tumor cells. First, we have characterized our TMP in terms of phenotypic analysis by flow cytometry. Our cells were absolutely viable and are represented by two different populations. The first that express the CD3, so they are T lymphocytes, exclusively composed by gamma delta T cells, both by the V delta 1 and V delta 2 population, whereas the subset that did not express the CD3 was represented by NK cells. We also found some B lymphocytes in our cell product, but the percentage of these cells was consistently lower than 3%, whereas monocytes were virtually undetectable. We have so cryopreserved our ATMP, and we have reanalyzed the cell product in terms of stability, so the maintenance of cell count, viability, and phenotype. But we have also performed some additional phenotypic analysis in order to understand and characterize our TMP for the differentiation status and for the presence of activation and exhaustion marker. Finally, we have analyzed our TMP for functional studies, and we have analyzed the cell proliferation and cytotoxicity against tumor cells, and in particular against leukemia cells, using the K562 cell line. Our TMP was stable, and until now we have validated a shelf life until 6 months. I have reported here the percentage of the different staining in red before freezing, and the panel showed the percentages after thaw. So as you can see here, our TMP was stable because maintained the gamma delta T cells and the NK cells inside also after freezing, and cells were viable. What about the activation marker? We decided to analyze gamma delta T lymphocytes and NK cells for the presence of killer activating receptor, and also for the presence of CD16, because it is important for RDCC functions. The killer receptor we have analyzed were NKG2A and D, NKP30, NKP44, and 46. And as you can see here, both gamma delta T lymphocytes and NK cells consistently express these killer activating markers. And gamma delta T cells were also analyzed for their differentiation. We used a double staining and flow cytometric analysis using CD27 and CD45 array markers in order to identify the 4 different main populations, so the effector memory, central memory naive, and terminally differentiated cells. And as you can see here, the large majority of gamma delta T cells were effector memory, so they are ready to kill the target cells. However, gamma delta T cells also express exhaustion marker, and for example, PIN3 and PD1. But the question is, gamma delta T cells are really exhausted from a functional point of view or not? So in order to address this issue, we have performed also some functional analysis, and we have analyzed the cell proliferation and cytotoxicity by the granulation assay. Our ATMP, our cryopreserved ATMP, has been cultured for 6 days in the presence of IL-2, and we have analyzed the CFCA dilution. And as you can see here, gamma delta T cells and both V delta 1 and V delta 2 subset may proliferate and perform in 6 days 4 division, whereas NK cells perform only 2 or 3 cell division. What about the cytotoxicity? We have analyzed the cytotoxic activity in terms of the granulation using the granulation assay. So gamma delta T cells and NK cells were the entire cell product was cultured in the presence of K562 cell lines, but also 2 different neuroblastoma cell lines that are the SKNAS and NSH. As you can see here, both gamma delta T lymphocytes and NK cells are cytotoxic. They are cytotoxic at the basal level, but cytotoxicity was significantly higher when cells were co-cultured in the presence of tumor cells. So in summary, we can say that we have really produced a ATMP that was safe in terms of sterility, absence of mycoplasma and endotoxin, and our product was composed of NK and T lymphocytes in different percentage. However, cells were consistently viable and cytotoxic. The presence of alpha-beta T lymphocytes and B cells were consistently very low, so under the 2.5%. In conclusion, we can say that our cell product is really pure, viable, composed of activated cells that are also cytotoxic and, importantly, stable after thaw. So we believe that our ATMP may be very useful to support leukemic pediatric patients after hematopoietic stem cell transplantation. Now we are working on to obtain the authorization that is under request to the regulatory authorities. In Italy is AIFA, and we are implementing the phase 1 clinical trial in collaboration with our clinician, and in particular Dr. Maura Faraci. And at the beginning, this ATMP will be used to infuse pediatric patients after alpha-beta T and CD19 B-cell depleted haploidentical hematopoietic stem cell transplantation to provide an immunological support and protect patients against tumor relapse and life-threatening infection. In fact, we are now analyzing also the ability of this cell product to work with an antiviral activity, and also we are addressing whether this kind of a TMP may be used also in MB patients after haplotransplantation. I would like to thank all scientists who collaborate with me and my group for the development of this ATMP, and in particular Dr. Fabio Morandi and Martina Della Lastra, who are involved not only in R&D but also in the QC lab. And thank you all for your attention.
If no other question, unless you guys have some, I am going to start. Can you comment a little bit more on the fold expansion that you have, and just to have a sense of how many cells at the end that you can think about having available for a patient? And since there is a zoledronic involved, Wu-delta-2 is probably the subset that is more abundant, but can you get anyway a good polyclonal product with the presence of Wu-delta-1 as well, or is more skewed towards the Wu-delta-2 population?
Okay, we have analyzed fold expansion of both the gamma delta T cells and NK cells, and what we found is that it depends on the cell product that we have validated. However, gamma delta T cells expanded from 20 to 50 fold, whereas NK cells expanded 2 or 3 times. So it is not so high expansion, but the good thing is that we maintained activated NK cells. Also because maybe in our context, gamma delta T cells and NK cells may cross-talk together. In terms of absolute number, it depends on the starting material. I mean, for example, we started from 3 billion cells, and we have obtained up to 500 of cells into our cell product. But in the phase 1 clinical trial, probably we will use an amount of cells that is feasible to use our TMP, also because we will treat pediatric patients, and this is a very good thing for us. Anyway, I believe that if we start from a large amount of cells at the beginning, we will produce more and more cells in our cell product. The Wu-delta-1 cells, we have analyzed only the percentage of V delta 1, but I don't know anything about the clonal expansion, each individual clonal expansion in our ATMP.